Mike. Welcome to CBE 411. In this video, we will use experiment to demonstrate the antibiotic effect of a Penicillium rubens strain, which is similar to the Penicillium rubens strain that Fleming discovered nearly 100 years ago. Before we start, I'd like to explain the aseptic technique we use, because we want to grow out only the strains that we are interested in. We have to protect them from other microorganisms in our environment, which also want to share the delicious food. Here are several aseptic techniques we use. Flame can create a cone of hot air above and around the bench to create a laminar airflow upward, so bacteria don't drop in this area. Gloves can protect the lab material from the microorganism on our hands, as well as protect us from the microorganism in our experiment. Autoclave is a machine that uses hot steam to reach 121 degrees Celsius and 1.5 atmosphere pressure, which can effectively kill all microorganisms. Keep in mind that it is the high temperature that kills bacteria instead of the high pressure. It won't be so easy to get a Penicillin rubens strain just by leaving an uncovered plate by the window, which is how Fleming obtained his strain. So we decided to buy a commercially available penicillin producing mold. Once we obtain an agar plate with P. rubens, we can inoculate the second agar plate. Here's how we do it. First, we prepare an agar plate with food that P. rubens love. Under an aseptic environment, we use an autoclave pipette tips or any other stereo tools with similar shape to cut a circular agar off the original agar plate. Then we use clean tweezers to pick up this small piece of agar, whose surface is full of the spores of P. rubens. Keep in mind that this pair of tweezers has been cleaned with ethanol and heated on flame to sterilize. Next, we leave this piece of agar upside down to ensure those spores are able to seed on the new plate of agar. Finally, we cover the plate and leave it in the room temperature, which is P. rubens' favorite temperature. In its favorite environment, P. rubens can grow quite rapidly. When it grows on sharpened agar plate at 25 degrees Celsius, its colony can cover the whole plate within 3 weeks after inoculation. In general, P. rubens love room temperature and humid environment. phenomenon Fleming observed is that P. rubens inhibit the growth of bacteria that are near its colony. We're going to use a similar experiment to demonstrate this. This time, we inoculate the LB agar with liquid P. rubens culture using stereo cotton swab. Here is the liquid culture of P. rubens. And here is the stereo cotton swab. We will dip the cotton swab in the P. rubens culture. And draw a stripe on the agar to allow P. rubens grow in this area. Two days later, we see P. rubens colony on the inoculated area. Then, we will use stereo cotton swab to inoculate the agar with 4 different bacteria strains, including 1 E. coli strain and 3 S. aureus strain. Again, we dip the cotton swab into E. coli culture and draw a stripe that is perpendicular to the P. rubens stripe. Keep in mind that once the swab touches P. rubens colony, it has spore of P. rubens on it. Do not use swab afterward, otherwise you will be spreading P. rubens all over the plate. Similarly, we inoculate S. aureus ATCC 25923, S. aureus RN4220, and S. aureus A325 to the different area on the agar plate. After leaving the covered plate at 25 degrees Celsius for 2 more days, we see this. All 4 bacteria grows fine in the first day, possibly because P. rubens is still growing and have not produced much penicillin. In next 2 days, we see S. aureus strains that are far from P. rubens grows well, but those close to P. rubens are lysed. However, the growth of E. coli strain are unaffected by P. rubens. This makes perfect sense because S. aureus is sensitive to penicillin, the chemical produced by P. rubens, while E. coli is not. Here the top one is E. coli. Second one is S. aureus ATCC 25923. Third one is S. aureus RN4220, and fourth one is S. aureus A325. Another experiment Fleming did was to show that the malt used by P. rubens also has antibiotic effect. Now we know that the active ingredient in the malt juice is penicillin. So we will use penicillin solutions to inhibit the growth of bacteria. We will use hockey stick to spread S. aureus culture on an agar plate. First, we use ethanol and flame to sterilize hockey stick. Then, we add the S. aureus onto the agar plate. Then we use the clean hockey stick to spread it. Next, we prepare a penicillin solution. Here is the penicillin solution. And we're gonna immerse the filter with penicillin solution and place the filter on the agar plate.
Finally, we leave the plate in 37 degrees Celsius incubator for overnight, which is the optimal temperature for S. aureus. We prepared 4 plates. 1 placebo, using filter with water instead of penicillin solution on the plate spread with S. aureus. And 3 plates with filters with penicillin solutions on the plates with 3 different S. aureus strains. Next morning, we see that placebo has no inhibition effect. And S. aureus that are too close to the filter with penicillin are unable to grow because the penicillin diffused.
Now we have shown that both P. rubens and the penicillin produced by P. rubens can effectively inhibit or even kill S. aureus. I hope this video can be helpful in understanding the discovery of penicillin. If you have any question regarding this video, please come to my office hour. Bye.